Great. Thanks for that introduction, Francis. It's indeed a pleasure to be here today, and as Francis said, it's an honor to give the opening keynote here. What I'm going to share with you today is how we're using bioinformatics in conjunction with genomics to try and see whether we can improve the healthcare that's provided to cancer patients in terms of more accurately understanding their disease, and therefore with a goal to make sure that they get the most appropriate therapeutics. If you take a flight west of here for about 5 hours, you end up in British Columbia. Population, if I remember correctly, around 5 million people. Most of them, as you can see here, kind of straddle the area a few miles north of the U.S. border. What's kind of unique, maybe about British Columbia Cancer Agency, particularly in comparison to say jurisdictions in the U.S., is that the BC Cancer Agency, a set of cancer hospitals, has the jurisdiction over all the cancer patients in British Columbia, in the sense that they're either all treated through the hospital or they're all registered. Their disease is registered through the hospital. So it gives us some insight into how cancer affects the population and how we as researchers can actually improve the healthcare for those cancer patients in that population. Now these numbers, I'm not going to kind of bore you with a lot of these numbers, except this is from 2012. So it's not incredibly up to date, but these numbers sometimes are hard to accrue. But for the population of BC, the BC Cancer Agency takes about 3,000 people employed to look after those patients. And then one of the numbers that I just want to draw your attention to is that it costs, in 2012, $184 million to buy the drugs for those patients. It's clearly well over $200 million at this point. And that would be just fine. You know, if we spent $220, $230 million on drugs for cancer patients and they worked and helped them, then that would be great. But the challenge is that most of that $184, $200 million actually goes and doesn't benefit those patients, and just actually reduces their quality of life by inflicting the toxicities that we're all aware of in terms of the fairly harsh and tough chemotherapeutics that cancer patients are given. So can we more accurately give those drugs? Can we make sure that the drugs are given to the people who will benefit and not to the people who won't benefit? Now, one of the things that we did a number of years ago, and this was really what inspired us to kind of keep going, is that a number of years ago, actually in 2008 or 2009, if I remember correctly, we had a patient come in, and we just acquired some of the latest Celexa, later to be Illumina DNA sequencing machines. But there was a patient who had a very rare cancer of the tongue. It's so rare, in fact, that the hospital had no established protocols to how to treat this tumor type. And so the clinician, basically said, well, at this point, we're just at the state of guessing. Could genomics actually have a role in helping us determine what was the most appropriate choice, therapeutic path, or at least give us some clues? So the BC Cancer Foundation actually, as a way of helping with genomics and research, actually helped to fund the sequencing of this tumor, one of the first tumors that was sequenced fully, and this is certainly one of the first. This is the first cancer tumor to be sequenced in real time for clinical decision making. Well, it cost about $200,000 at the time. But what we were able to do is we were able to, with the transcriptome, if the mouse is, with the transcriptome here, for instance, and the mutations that we observed in the tumor, we were quite quickly were able to see that the oncogene RET was very highly expressed within this tumor, much higher than we even see now in more common tumors. And so that immediately gave us, some therapeutic insight. And this actually was able to allow the clinician to prescribe a RET inhibitor, and actually the patient went into remission for a number of months. So that was our first example of using genomics in clinical real time for clinical decision making, and in this case had clinical benefit. So in 2012 we started with the idea as well, why don't we come up with a strategy, a pilot project where we could expand this idea. And I think that we all know that the future is the tumors are going to be very well molecularly characterized, that we are going to be interrogating their genomes and transcriptomes much more in the future than we are in the past. So this to us seems like a very obvious route to go forward with. However, there's things that we can do in a research context, but it's much harder to do it in a clinical context where we have turnaround times, and we have to worry about schedules and biopsies and all those kind of things. So the first part of our project was really to understand, in a cancer care center, can we actually sequence human genomes at a pace which would actually be useful for those patients? Can we determine what is required? We still need 3 genomes. We need the tumor and the normal, and conceivably a diagnostic tumor that was sequenced that was taken when the patient was first diagnosed. We might need a transcriptome. I think that turns out to be true. What are the bottlenecks in that process? What kind of analytic pipelines do we have to set up to do this routinely? And how do we communicate the results? The clinicians haven't been trained in genetics. They haven't necessarily thought in the context of pathways and mutations in the same way that we as geneticists and genomicists have been thinking. So clearly there was a culture that needed to be educated in how to interpret these results as well. So we worry about doing this in a timely fashion, and also how often do we find things that are actually useful for the patient. And maybe it didn't matter early on, because I think we would understand or expect that as time goes on, as we learn more and more about tumors as a community, that we will have more and more insights from the tumors. So this is the idea of the pipeline that we set up. We have the consultation. We need the appropriate sample acquisition. We need to understand the kind of biopsies we would need, and these may need to be of a different quality or a different type than the kind of biopsy that a pathologist might need just to make a diagnosis. We need to have a sequencing strategy that's going to find the somatic mutations that we need to. And we need, again, the analytics, the bioinformatics, to interpret the sequencing results. We need to turn that into something that's actually interpretable by humans who aren't necessarily bioinformaticians, and then we would take that to a multidisciplinary tumor board where a discussion would take place in terms of what do those mutations or do those results from that analysis mean to the patient's care. And then ultimately those would then go to the patient, who would then consult with the oncologist, and the oncologist would indicate what path the sequencing has determined, if any. So that's the kind of pipeline, a very 30,000 feet pipeline of overview of the pipeline that we have. Okay. When we started this, we didn't want to go in and say, well, let's just do colon, or let's just do breast cancer, although that was discussed quite a bit. The idea would be to take patients who we can have an accessible biopsy, and we could learn about all different tumor types. And this is kind of a breakdown of say, the first 388 cases that we did. So there's all different tumor types that are represented, and it basically breaks down in terms of how common tumors types are in the population. So breast and colon, for instance, are the two most numerous cancers because those are pretty high up in the frequency in our population. Okay. So just to give you an idea in terms of how we apply this to treatment planning, we take 100 cases, we end up with about 78 with can successfully go through the biopsy process, and we actually get material that's useful for an analysis. And I should say that part of this is that our initial approval was for very sick patients who were completely out of options. So many people were accrued into the study, but were too sick ultimately to have a biopsy. Of the 78 that we sequenced, we found that about 65 were informative in the sense that there was some insight whether not to give a drug or this pathway is upregulated, and we could, for instance, have a trial of some kind that the patient could be enrolled on. So about 55 we determined were actionable. And unfortunately, of course, with a very sick population, about 13 were too sick at that point to receive treatment. But 34 were treated, and we were able to get 14 patients actually had improvement. So actually, we think that's actually pretty good based on the fact that this is a very sick population. 100% of these were metastatic patients. What we're discovering, of course, is a lot of established alleles, which is good because that allows us to partition those patients into kind of known actionable categories or understand the kind of pathways that are being changed or mitigated in those patients. But we're also seeing a lot of novel coding mutations in these patients, and whether they're passengers or drivers, I think, is yet to be seen. Most of the data coming out of, say, TCGA and the large consortia focus on primary tumors where these are metastatic, and so some of these are going to have a role in metastases. Some of them are going to have a role in accruing resistance to drugs that these patients had already had, and some of them, of course, may simply be passengers. But at least we're starting to build up a picture of the kind of mutations that we're seeing in these patients. So what we did after that, some lessons were learned, and we went on to start version 2, which is ongoing. That actually has allowed us to get REB approval, so Research Ethics Board's approval, but not necessarily the funding for 5,000 cases. But that's the goal, is to move forward. And in a slightly different patient population. So instead of getting patients who are completely out of options and with very advanced metastatic disease, we'll be getting their genomes done at a much earlier stage, which gives us a much longer time to follow those patients and come up with a much better therapeutic insight, or wait until they've finished their current therapeutics before we start to necessarily have to change them based on the DNA results. But to continue to sequence the genomes, the normal genome and the tumor genome, and the transcriptome. Okay. So in terms of the informatics, we've developed a pipeline, as everyone does, of course, in terms of how you want to passage the data, through analysis. I think one of the challenges here is that may not be totally appreciated. Is really, how do you do this in a clinical scenario, right? We have to validate these tools. We can't just say we're going to update the aligner version today. That involves quite a bit of validation and testing to make sure that aligner is doing exactly what it was doing previously. So each of these steps has a validation protocol. And so to actually change them is actually non-trivial and actually quite tough, which again is quite a headache for me and my group. But this is what happens when you need to do things in a clinical environment where everything is very reproducible. we can follow and make sure that each step has completed correctly. But what we're doing for the most part, obviously, we're doing SNV calling, where we're looking for somatic changes that are in the tumor that aren't in that weren't in the germline, which is typically derived from blood. We're looking at copy number changes, loss of heterozygosity. We're doing an assembly, a de novo assembly of the genome to give us a little bit more insight into the structural rearrangements that have accrued and the breakpoints of those structural rearrangements. And we do a lot of assembly on the transcriptome, and hopefully if we see variations in the genome, they'll be replicated where appropriate within the transcriptome. And of course, then we want to quantitate the relative expression of the genes within the tumor and identify any novel or known isoforms within the transcriptome as well. So that gives us a lot of data to look at in terms of the molecular biology of the tumor. And then a large part of what we're focusing on then is trying to come up with a drug or at least a pathway annotation where we can say this is the pathway that's changed in the tumor. This is the pathway that we should be thinking about therapeutically targeting. And that is the challenge. If you guys want a challenge here, I mean, I think this is where machine learning or other kind of approaches are really needed to kind of help speed up the hotspots in the pathways that we need to be looking at. Quite a bit of time again is spent when we find alleles of interest, we have to go back into the literature and understand a little bit more about what they mean. And again, that isn't really automated at the moment. That involves a lot of people reading about the changes that they've observed, reading about the genes that are mutated to get some kind of insight. Now, of course, we can record then those insights, and hopefully they'll be useful for subsequent patients with similar mutations. But this is an example of when we've done all this analysis. This is an example of a kind of the insight that we would build up. Here's an example of a breast cancer, for instance, and so we would build up that here, for instance, we've got a fusion. We're seeing expression now of this gene, which is a known oncogene in the 100th percentile. We've got a gain of function mutation here. We've got a copy number gain of this gene here. So we start to build up what we think is the mitigated or the changed pathways, and then that allows us to come up with the most appropriate drugs. And again, that's currently done by a human in terms of going through and checking that everything makes sense in the context of the known literature. But in this case, for instance, just identifying the fact that we believe that this is the driving event here, and there are drugs that can target it, is actually now helpful for the patient. And this certainly wouldn't have been known before we did this kind of molecular analysis. So this is the output that we want, right? We want to be able to go to the clinician and say, we've solved this one in terms of the genes and pathways that are driving this tumor, and here are some suggestions for drugs really based on hard data and the molecular biology of the tumor. So currently, this slide says 4 cases per week. We're probably up to about 7 cases per week now. And the idea is to kind of drive these through and ultimately come up with what we call a report, which provides all the salient information in terms of what the clinician or pathologist actually needs to know about this tumor, and what the important mutations are and what the important genes that are being expressed aberrantly in this tumor are. So this has taken us, this is quite a lot of work to actually get to this stage. A lot of what we're doing is trying to make sure that when we capture information that we're not having to go and find it again. So we've established internally what we call a knowledge base, which is basically all the events that we should be looking at that or looking for, that indicate that there's a particular drug or a clinical path that should be followed when we observe that. And we're actually doing this now in collaboration with Washington University with their CIVIC database, which aims to do many of those things. So many of the actionable mutations that we find, we're actually passing those into the CIVIC database, which is actually a freely accessible, open database of actionable items for human cancer. But the process that we've outlined actually involves a lot of people. So just to outline that this isn't a trivial thing. It takes a lot of time. So we've got about 15 informatics people that are worrying about sequencing essentially 21 genome equivalents per week. There's a number of other people working on preparing the libraries, doing and running the DNA sequencing machines, all those kind of things. There's radiologists and medical oncologists that are involved in analyzing the results, and the pathologists, of course, and then there's people who actually have to do the biopsies as well. So this is now as a fairly hefty undertaking to try and do this in a routine manner. But let me give you some examples now of the kind of things that we've been finding. So for instance, here's an example of an ovarian cancer, and of course it just would look like any commonly observed ovarian cancer from the pathology perspective. But what did the molecular biology tell us? And so, for instance, what we were able to find in the genome when we do the assembly of the genome is that there's a large number of translocations, if we just look at this kind of Circos type figure. And there's also translocations that are supported by the transcriptome. And one of these is a patched GLI1 translocation. Now, GLI1 had previously been known to be involved in oncogenic fusions, but not actually linked to the patched gene. So, for instance, in pericytoma, people have observed ACTB GLI1 fusions, where you've got the first two exons of ACTB fused to exons of GLI1. And so it's a common kind of mechanism of a promoter swap. You basically put GLI1 under the control of another gene, and that will increase its expression, and that's how you get a kind of an oncogenic driving event. So in this tumor, we're seeing a similar GLI1 fusion, but with a different driving event. And in this case, this was patched 1. So we were observing perturbed expression. But what was interesting is that the molecular biology tells us that actually GLI1 actually positively controls the patched 1 promoter. So you've got this out-of-control feedback loop now, where GLI1 is regulating the expression of patched itself. Okay, so so we would predict now a crazy amount of GLI1 being expressed in this patient, and we're thinking then this would be a driving event. So if we go look at the expression now, for instance, so if we look at a control genome, so not the one with the fusion, this is the kind of expression that you get for patch 1, and this is the kind of expression you get for GLI1 for each of the exons, with the y-axis being the RPKM. Now we can look into this particular ovarian cancer, and we can look at the expression of our fusion, bearing in mind that we should be seeing only a subset of the exons being expressed more highly. So we see exactly that. We're expecting a high level of exon 1 from patched 1 because of the positive feedback. So we see a very high level of exon 1 in this tumor compared to the control that we're just using for comparison. And for GLI1, what we're serving is we see normal expression of exons 1 to 5 because they're not involved in the fusion, but then we get huge expression of exons 6 and subsequent. So this really showed us that not only were we observing the fusion, but the expression data was showing us that there's a huge amount of these transcripts actually here in this tumor. And just drive this home, these are very different axes, right? I mean, this RPKM here for the patch 1 GLI fusion here is 1.5, and this is 150. So the scalia is 100 times that of the control. So we're seeing this for the first time. We're seeing this fusion in this ovarian cancer. We don't see it in other ovarian cancers, and I think this is a theme too, is that many of these cancers have unique properties that aren't common, and that's possibly why we haven't cured cancer, is because all of these tumors have very unique aspects to their molecular biology, and yet will share common ones too. But it's these outliers that seem to accrue fairly commonly. So this was able to put this tumor, onto a pathway, and we were able to show that it was the hedgehog pathway, or we believe then that it's this fusion here that's activating GLI1. It's this self-activation of patched 1 that's really forcing this growth pathway, the hedgehog pathway, and is a driving event for that tumor. And people are developing hedgehog pathway inhibitors, and so this again is something that this patient theoretically could go on, once those inhibitors are developed. Another example, in this case, January 2015, an adenocarcinoma of the vulva, and actually had responded a little bit to standard carboplatin Taxol type therapeutics, but actually rapidly went to metastasis. There's really no standard treatments in mind at this point. They'd exhausted the current protocols. But when we did the analysis, part of the quality control is to compare it to all the other known tumors in terms of the expression. So we didn't find this necessarily to look like adenocarcinoma of the vulva. It actually looked like a breast cancer when we compare it to the expression profiles. The highest correlation that we had was with breast cancers. So that made us thinking, well, maybe the diagnosis is wrong. And this is actually something that then we can go back to the biology. So when humans develop in the embryo, just like actually most mammals, the mammary tissue actually extends across the entire abdomen, and it's in humans and other mammals where that actually degrades and it only forms in a certain place. And if you have dogs or cats, you'll know that the breast tissue actually extends the entire length of the abdomen. So what happens here is that in humans, we have what we call a residual milk line of vestigial breast tissue, and actually that's what the tumor was. It was a kind of a vicarious breast cancer. And this is actually very important to the patient, because then the patient actually has now available to them Herceptin, for instance, which we were able to show that this was not only a breast cancer, but a HER2-positive breast cancer. And so actually the re-diagnosis was a very positive outcome for that patient. And in fact, there's a number of fairly effective therapies now for HER2-positive breast cancers. Another interesting case, 60-year-old male, lifelong never smoker, and diagnosed with lung cancer. What we did is, from January 2013, had ongoing progressive disease, had tested for the standard mutations that would indicate a certain drug that would be useful, EGFR and ALK. But actually, even though they'd failed the standard pathology test for an ALK fusion, our de novo assembly actually found one. And it was because it's actually a very complex rearrangement. And a de novo assembly was able to put back the ALK fusion correctly, whereas the standard pathology test just simply failed. So it's an example of when you do a complete genomic analysis on a tumor, you're actually independently and comprehensively doing kind of all the known tests at the same time, and conceivably maybe in a more accurate way than a PCR test, for instance, that's done on some biopsy tissue. So we were able to show and recreate a much more complex fusion that the standard pathology test missed, and was very important for that patient because we were able to show that subsequently from the transcriptome as well that the ALK gene, which is fusion interrupted or involved, now put expression of that in the 99th percentile. So in the top 1% of expressors of this gene. And that was very important for this patient who was actually only a few days from death when we actually were able to find that. So we were able to give the drug crizotinib immediately once we found that result. And if you look here, for instance, with fairly large tumors on September 14th, we were able to scan then September 25th, and they basically had disappeared. So again, very, very accurately being able to understand the molecular biology of the tumors was really, really important for that patient. One of my favorite stories, again, a 40. Another woman with lung cancer, metastatic lung cancer, of course, it had standard chemotherapy with some response. Lung cancers typically progress fairly aggressively. Again, we were able to find a novel fusion. No one had seen this in lung cancer before, of the gene NRG1 with a partner SDC4. SDC4 probably only having the role of lending its promoter to aberrantly express this oncogene. This SDC4 also contains a single peptide at the end terminus, where we think it's now delivering NRG to a place where it can actually activate ERB kinases, also known as HER kinases. And so what this has allowed us to do was to build up another pathway with this gene fusion at the top, and basically the expression that we're now seeing of this gene in the 91st percentile showed that a pattern where we think it's basically being oncogenic through the expression of ERB3 and ERB4. And then there were other copy number changes, like this gene here in the pathway has an amplification, and so does this one, and so does this one. So this gave us some pretty good clues that this was then the pathway that was driving this patient's tumor. And so we were able to give an ERB4 inhibitor called afatinib, and this was again able to have a positive effect on that patient's tumor. So previously, of course, we would have known nothing about NRG1 driving this patient's tumor, but we can see on July 16th, we can see this tumor here, and by September the 10th, effectively a large amount of that mass had disappeared. So again, very insightful. We can start to learn about what these fusions mean, and hopefully what they will mean for other patients in the future as well. Another interesting case, very interesting in this case, a 67-year-old female with colorectal cancer, standard 5-FU platinum type therapy, was metastatic, was actually on palliative care by the time we were able to do our analysis. Had a number of tumors, including one that was causing a lot of pain and affecting her spine. And so in June 2014, we had the biopsy. What was kind of interesting about this tumor is that the number of protein coding SNVs, single nucleotide variants, was about 1,500. So maybe 20 times more that you'd see in a tumor. Likewise for frame shifts. There was a vast number of oncogenic mutations that we were observing. Gain of function in variants of unknown significance in PIK3CA, probably a gain of function in GNAS, loss of function of a number of tumor suppressors, and whether they were all driving or participating in that tumor at that time, or a reflection of the large number of mutations that the tumor had, is not 100% clear. But what we did notice, that there were two genes called FOS and JUN, which are also on an oncogenic axis here. And we'd never seen the kind of expression values that we've seen for those genes in any other tumor. Consequently, especially in combination, and especially for FOS. So that kind of tweaked our attention. It says, why is this tumor in particular expressing these genes at such high levels? And it's probably because the tumor needs it, and they're probably driven to such high expressions because they are driving the tumor. And we said, well, does that represent a therapeutic path? So FOS and JUN actually live on the angiotensin pathway, which is typically involved in regulating blood pressure, not necessarily cell growth. So that was kind of interesting in the sense that these live here on this well-established pathway, and they're under control of the angiotensin receptor. And that's fairly commonly targeted by blood pressure medications that are widely prescribed within the human population. So it seemed a bit weird, but is that something that we could try? And the patient was actually so sick that we couldn't really give any chemotherapeutics at that point. But the clinician thought, well, why don't we just give blood pressure medication? Because that's what the data is telling us, that this is a pathway that seems to be playing a role in the patient's tumor. And so what we can show is that pre-treatment, here's the metastasis around the spine, which I believe is the site which was chosen for biopsy as well. But there's a number of metastatic lesions throughout the body as well. But within a few weeks of just taking a simple blood pressure medication, that's what happened. So we can go back and forth, and the tumor disappeared. Now, we've never seen another tumor like this in terms of that particular molecular profile, but this is a drug that costs 50 cents a day, not $10,000 a month. But importantly, it worked, and importantly, it has no toxicity. We don't know whether it would work on any other patients, but it worked for this patient, and that's kind of enough because it was, it was the molecular biology and the bioinformatic analysis that actually led to that, and obviously our insights into the molecular biology can be therefore quite powerful. So someone went from being on palliative care to actually spending the next summer in Greece. Now, so for almost a year, the tumor had disappeared completely. So she was essentially cancer-free. She had no detectable tumor at all. And then 1 year later, we started to see a small growth. So we re-biopsied that. And this is fairly recent, so we haven't done a huge amount with this. It's still very small. But what was kind of interesting here was that the mutational burden had gone up. So the first biopsy that we did, we also did the archival specimen. I won't talk about that. But the biopsy that we used to inform us of that therapeutic had 2,300 protein coding mutations. The biopsy now has accrued even more at 2,500 protein coding mutations. So it's certainly got a mutational burden, and whatever it's the 200 mutations it's accrued over the last year as it's come back. I mean, that's about 3 normal tumors worth. So it's rapidly mutating. But the first biopsy and the second biopsy, they share about 1,700. No, actually, sorry, they share 1,700 plus 533 mutations, right? So it's clearly very, very similar tumors that has recurred. But this actually does give us some therapeutic insight. So PD-L1, I don't know how many of you are familiar with immunotherapy, but as a tumor accrues more and more protein-coding mutations, the more immunogenic it becomes. And so what we also saw is a shift in the expression of PD-L1, which is something that protects the tumor against the immune system when it's very immunogenic. Went from the 55th percentile up to the 89th percentile. So actually that gives us now a fairly good therapeutic path, because now we can give a PD-L1 inhibitor. Because this is one of the most mutated tumors I've ever seen, I think it's got a pretty good chance of working. What was interesting is that, and when we saw that the tumor was coming back, my first impression was that the expression of Jun and Phos must have actually gone up even more to counter the effect of the drug. But actually they went down. So Jun actually went from being in the 100th percentile, essentially the most highest level of Jun we'd ever seen, actually going down to the 69th. And Phos went from the 97th percentile down to the 72nd. So it's interesting. These were the 2 top changes that we saw between the first tumor and the second relapsed tumor, right? So I think this lends us evidence that it really was Jun and Phos that we were targeting, because the relapsed tumor, those are basically the only things that have changed. So we're not only being able to look at these tumors in terms of how we might treat them, but we're now able to look at their evolution over time as they respond and potentially become resistant to certain therapeutics. Okay. And then in the last 2 or 3 slides, I just want to kind of say something about mutational signatures, because these are something that we can get out of the tumors fairly easily. So one of the things that we were looking at is, can mutational signatures be of use? Certainly for some tumors, the clinicians come with tumors where we don't know for certain where they actually arose. It's a metastatic lesion, but doesn't look like, for instance, it arose in the liver, or as a liver cancer, but it's a metastatic tumor that's now spread to the liver, but we don't know where it actually started. So we get sometimes these tumors, which are called primary of unknown origin. When we look at the different mutational signatures, we find that this is involved in smoking. We only see smoking signatures essentially in the lung and the associated organs like the esophagus. So I think it's pretty clear, for instance, that we were able to diagnose that tumor as a lung cancer just purely based on the mutational signature. And again, very important for the patient because now that opens up approved therapeutics for that tumor type. So this is the mutational spectrum that we see of the tumor, and these are the 3 signatures that we detected within it. And as you can see here, this is the tobacco signature, and we have a fairly strong linkage with a tobacco signature. So we were able to quickly draw a line under where that tumor actually came from. Another tumor of unknown origin, but you can quite quickly take the UV signature that occurs basically when you get exposed to sunlight. This is the signature here. This is the signature of the tumor. Fairly quickly can be diagnosed as a melanoma. And again, really important now that there's some very effective drugs that can be used against melanoma. So again, very important for that patient. We can look at breast cancers, for instance. When we look at all the breast cancers that we've studied, we come up with 6 signatures the kinds of mutations that accrue within those tumors. The most common one being defects in BRCA1, BRCA2 genes, which are DNA repair genes. Again, this is very important in the sense that if you do have a BRCA1 or a BRCA2 signature, you're going to respond fairly well, actually, to a platinum-based therapeutic. If you don't have the signature, then you're just going to get toxicity. So what we were able to do, and actually this is a very busy slide, and I think it's my last slide, is really take and calibrate the signature. And what we were able to show is that for breast cancer patients, the higher the signature we observe, the better you will respond to platinum, right? The number of days you will remain on platinum is directly linked to the strength of the signature. And what we were able to show is that there were some patients who actually didn't have observed BRCA1, BRCA2 mutations, but actually had the signature. And we were able to show that if they don't have the mutations but still have the signature, they respond to platinum. And that's important because previously those patients would never have received platinum. But now we can show that they should have platinum based on purely on the mutational signature. So we were able to show that mutational signature is actually a better biomarker of platinum sensitivity than the BRCA1 or BRCA2 mutations themselves. So this again is something that we can quickly get into clinical practice, at least in British Columbia. Okay, so just to conclude, we are showing that it's feasible to do genomic analysis on tumors. It's feasible. It's still a lot of work. How scalable it is another question. Can we do it fast enough? Well, actually, if you take patients at the right time in their disease, actually there isn't a huge amount of urgency. So that's really what we have to consider. What's the frequency of actionable results? I think it's actually fairly reasonable, and it's only going to get higher. And one of the challenges, of course, that we have in British Columbia is that there's often times where we know that there's a drug that will help that patient, but the health system isn't going to fund that drug for that patient because it's not necessarily approved for that tumor type at that stage. So that's one of the challenges, right? Because many drugs are based on having a disease type, like melanoma or lung or whatever, but not a molecular type. And so that's hard to see how that can be solved quickly. And I realize from many examples I've given you, for instance, there are no controls. Many of the treatments that we ultimately give to patients weren't our first choice. I gave you reasons for that just previously. Is it cost effective? And then we'd have to say, well, what would be the comparison? And at least in the first study, metastatic patients were a very tough population in which to work. Anyway, I'll just stop there. Here are all the people that are involved. The PIs, Mark O'Mara and from the genomic side, and Janessa Laskin from the clinical side, and then all these other people have touched the data and have contributed in some way or another. So with that, I'd like to thank you for all your attention. Right. Well, we can't establish that with. No. I'll. Okay. I will attempt to repeat the question. So I think what you're saying is, when with the blood pressure medication, we proposed that it was working for the angiotensin pathway. But although it worked, how do we actually know that it was working in that particular way? And my answer is, well, we don't. Not at this point. We're spending our time trying to follow up on the molecular biology in the lab and in cell lines to try and really get to grips with what was a very profound response to that patient. The response clearly was associated with the prescribing of that medication, but how it actually works, very, very interesting. Yeah, we're certainly following up on that, but we can't be sure at the moment. So from the time that we identified the patient could benefit from being on the blood pressure medication, they remained on it. Yeah.